Good afternoon, everyone. Please be welcome to the colloquium named after the printing of Colonia, Dignidad for the forensic science. My name is Rodrigo Núñez. I am a professor of the Department of Filosofía at the Universidad Católica del Maule, and on behalf of the university and also of Mrs. Mirna Troncoso, coordinator of the Agrupación de Detenidos Desaparecidos del Maule, I would like to greet you and to thank all of you, your presence today in this panel. In this second panel of discussion, that is the scientific analysis applicable in Colonia Dignidad for search of the truth, gathering a group of international experts in the forensic anthropology, legal medicine, microbiology, and the science of soil. As we can see, this is a very scientific group that we would like to welcome. First, I would like to greet Mr. Luis Fondebride, forensic anthropologist, founder of the Argentinian team of forensic anthropology, to whom we have known because of his contribution to different cases of national interest, Patio 29. And thanks, Luis, for being with us and also for being the moderator of this table. I'd like to say welcome to Sandra Emmons of University of Colorado. Thanks for your presence. Her recent studies are very interesting for us, especially those related to the preservation of the human DNA on the soil. Thank you, Alexandra, for being with us. Also, we want to welcome Jennifer Dubrin. You are Associate Professor of Engineering in Biosystem and Science of Soils at the University of Tennessee. Thank you for sharing your time with us here. And also, I would like to welcome Ivan Cáceres. He knows firsthand this study. Good afternoon. Thanks for being here with us. Briefly, I would like to finish on behalf of the Universidad Católica del Maule and the Agrupación de Detenidos Desaparecidos in order to be connected with the spirit of the first roundtable that we had today in the morning. That is a human sciences roundtable with the participation of philosophers, historians, and other disciplines in order to have a point of view the people who have suffered this horror in the area close to Parral in the south area of Chile, to their relatives to whom we have a human commitment and closeness, and also an ethical commitment. This morning we have discussed about the category of the prints, the concept as a possibility to reconstruct the memory of the history, but also as a horizon of ethical and political responsibility for those who are committed to cooperate from our knowledge in order to have progress in justice. And these are my end words for the second session. Thanks to Jorge González and Verónica Madariaga. They are the simultaneous interpreters today who have cooperated with us. And we beg all of you, for all those who are talking, to pay attention and to speak as slowly as possible for the interpretation. I think that is desirable at the time that we can make some pauses for the interpretation. So Luis Fondebride will start the session for the coordination of the next stages. Thank you very much for your words. Good afternoon, everyone. It's a pleasure to be here with you, sharing this table with you, with colleagues from the United States and Chile, in order to have some answers to all those relatives who want to know what happened with their beloved people. Latin America during the last 30 years had had a long history with steps backs and forward in order to find justice, the truth. All the countries in South America, especially in the southern area, after the military dictatorships have started through Comisiones de la Verdad, through the use of justice and other mechanisms to know what happens with the missing people, if they are alive, dead, and also demand that those people responsible for the deaths face the justice. No doubt Latin America is an example, despite we have a lot to continue, but also there is a merit that we have achieved a lot during these years, and we owe that to what is the human rights movement. There are many thousands of people and common citizens from different eras of the society who demand to know what happened, to demanding this to the governments. We know the processes in Central America, Peru, Colombia, Argentina, Uruguay, Chile, and we know what has happened in these countries. In case of Chile, especially, we have the case of Colonia Dignidad. This is a special case, not only in Latin America, but also in the whole world. It was a place created in 1961, and it has been present in not only in the dictatorship, but also in democracy. And so far, we don't know where the bodies that, because of different witnesses, we know that those people died there. The science has been working in different occasions. There is a judge in charge of the investigation, but also what happened in the cemetery of Patio 29, Area 29. Chile has started some efforts to provide a new turnkey to these investigations, as we did with the forensic uh, meetings, supporting the local scientists, trying to find the right answers to complex problems. For instance, the case of the Patio 29, Area 29 of the cemetery. By 2006, I was part of the International Commission of Experts in charge of the case. Then we have the case of Pablo Neruda, the poet, Victor Jara, and that mechanism, with international sciences and Chilean science working together, had a very good fruit. What's most important was the credibility and transparency, especially for the relatives of the victims here in Chile. And in case of Colonia Dignidad, despite we have had some research, once again, the relatives are demanding that to have some organizations, people from abroad, and all together with scientists here in Chile may think about the challenges that we have with Colonia Dignidad. This is a very complex case, not only because of the time passed, but also because of the specific features of this place that is so huge. In order to scientifically discuss, but without losing that sense of the pain those relatives feel, we have gathered three scientists, one from Chile and two from abroad, who in their own fields, they are leaders in the world in these areas. We are going to have first Ivan Cáceres, Chilean archaeologist, then Jennifer de Bruin, microbiologist, biologist, and a specialist of decomposition of carcasses. She works in the uh, famous farm in Tennessee, and also uh, Alex. She's going to be in charge of the speech regarding DNAs of soils. Okay, we will start with Ivan. We will explain the idea. Every speaker will have 20 minutes with a PowerPoint. There will be interpretation from English into Spanish and vice versa. And then we are going to start with the roundtable of questions and answers. That will be done with the chat in YouTube and Facebook. That will be coordinated by Luis Narvaez, and then this is how we are going to have the evolution of the activity. First speaker is Iván Cáceres from the Universidad de Chile, and he finished his career in 1996. He has participated in Chile in different areas, trying to find the missing people. He was a member of the group of anthropologists, the GAF, and in this case, in Colonia Dignidad from 2005 to 2018, he has been in charge of the archaeological diggings in different areas of Colonia Dignidad. Ivan, please, the floor is yours. Thank you. Thanks for the invitation. And I would like to share a PowerPoint. Let's see. Can you see it? Please let me know if you can see it. Can you see that? Not yet. Okay. I think I uploaded that. Just let me know if we have some problems with that. Maybe you can upload it that. You have to share the screen.
Ahí está viniendo.
Okay, that's it. We can see that. Now it's open. You have to open your presentation.
Todavía no.
Not yet. You're sharing your screen, but of the folder. You have to double click on the PowerPoint.
No lo ven, ¿cierto?
Can you see it?
Todavía no.
Not yet.
¿La presentación la tenés abierta abajo?
The presentation is open. Where?
A ver, que tengo un problema que aparece.
Let me see. I think I have a problem.
Un problema ahí, pero
Tenés que tratar de cambiar de pantalla, Iván.
sí, sí.
You have to change your screen.
Bueno,
Luis Narváez, ¿podés poner la presentación que mandó Iván?
Luis en este momento está resolviendo, creo. Nos dijo que iba a proyectar la presentación.
Dale modo de presentación, Iván.
¿Ahí se ve?
No, el tercer circulito abajo a la izquierda. Si moves el cursor para abajo, para abajo, para abajo.
Abajo a la
Abajo.
izquierda.
Abajo de todo a la izquierda. Ahí. Se va el cursor abajo de todo.
¿Me escuchas o no?
Sí, yo te escucho, pero lleva el cursor abajo de todo al modo presentación.
Sí, sí, pero no me parece. Aquí dice compartir pantalla, ahí estoy. Algo pasa ahí.
Sí.
Yo te veo, Luis, no sé si ustedes me ven a mí.
Estamos viendo la presentación, pero está
Sí.
toda junta. Tenés que ponerle
We can
abajo.
see the presentation, but we can see it with all the screen. You have to put it into full screen. Yeah, click on that.
Sí,
Dale clic a ese.
ya.
Click on that. Double click.
¿Y qué me dice? Me dice que tengo que entrar con Gmail.
Igual se ve la presentación, ¿eh? Si querés comenzar, está bien.
Anyway, we can see the presentation that way, but I don't see the presentation. That's a problem.
No sé si lo pueden ayudar alguno de la universidad.
Someone from the university, can you help me out?
Dígame.
Luis está compartiendo su pantalla.
Luis is sharing his screen with your presentation. If you open Zoom
Donde está haciendo el coloquio, va a ver
with your presentation, you will see
presentación en PowerPoint de manera
that
que
your
usted puede
presentation
tomarlo
is in
y Luis puede
PowerPoint, so
usarlo.
you can speak, and Luis is going to
Ya, pero
advance
no
the
estoy
pages.
viendo aquí
But I
la
don't see my
presentación.
presentation. I see.
No me resulta, así que no sé qué es lo que pasó. ¿Qué página tienes
I don't see
ahora,
my
Luis?
presentation. What's your page, Luis?
Luis había compartido recién pantalla con su presentación.
We had shared the screen with your presentation, so you can see it directly from Zoom
Sí, claro, yo estoy
with the
viéndolos a
panel.
todos ustedes.
I can see you
Yo abro
all,
la
but
presentación,
I
le
don't
doy
see
a
the
compartir
presentation.
pantalla.
I put share screen. I click on that.
Ahí sí, ahí estamos viendo su PowerPoint.
Okay, there are now we can see your presentation. That's okay. That's
Ya,
perfect.
bueno.
Okay,
Bueno, ya están viendo, ¿cierto?
now you can see it.
Bueno, démosle
Okay,
entonces.
let's start.
La Colonia Dignidad
Colonia
es
Dignidad. What is the
se ubica en el
background?
centro-sur de
It's
Chile,
located in the central
a unos
south part of
350
Chile,
kilómetros de
350
Santiago, de
kilometers
donde
from
están viendo
Santiago,
ustedes.
south from Santiago.
Y como lo había dicho Luis,
And as Luis said,
corresponde al enclave alemán,
this
donde
is a
todos los
German
alemanes
settlement of settlers,
prácticamente como una secta desde el
and
inicio de los años
it's
60.
sort of a sect from the
Después del
1960s.
golpe de Estado en Chile en
After the coup
1973,
d'etat
se
in
usaron los arrestos de
1973 in
delincuentes
Chile,
como un
it
centro
was
de
used
detención y
by the
de detención
dictatorship
política.
as a center for
Se estima que más
detention
de 30
of
detenidos políticos
political
fueron
opposers.
ejecutados al interior de la
More
colonia.
than
Esta
30
cifra
people
hay que
were
tomarla con cierta
executed
cautela porque algunos hablan hasta
in
de
this
100 personas.
area. Some people say 100
Y
people.
las
We
víctimas
don't know that. We have to be very
provenientes
cautious. And the
de
victims
las
were
áreas
coming
vecinas
from
de
the
Concepción
surrounding
a
areas,
Valdivia,
even
de
Concepcion,
Santiago,
another
donde tienen
city
cuarteles
southern from Santiago, and
¿ya?
also from other
En
areas in Santiago.
1978,
In
5 años después del
1978,
golpe de
5 years after the coup
cuando los
d'etat,
hallazgos en Chile
and with the
de
findings in
forma
Chile
fortuita o por
at
denuncias
random or because of
de testigos
some
desaparecidos
witnesses of some victims
en
of
el
the
centro de
dictatorship
Santiago y en el sur,
in Santiago and the south, there was evidence that the missing people had been undigged in several places, public and private places. So the military commander in 1978, because of these findings, made what was called the Operation Removal of TBs. This operation was simply the illegal undigging of all the victims in order to make them disappear and to lose any debris. Obviously, the TBs in this case were the victims. And in general, this illegal undigging, this operation had a destination of the sea. The bodies were sent and dropped into the sea. In all Chile, in general, most of these illegal activities were made by the military people, except in Colonia Dignidad, because this activity was made by the settlers, supported by the military. And as we said, that in the other cities of Chile, the bodies of the missing people that were illegally undigged were dropped into the sea. In case of Colonia Dignidad, the witnesses said that the bodies of the victims that they undigged were burned, and the ashes were dropped into the Rio Perquelauquén. That's the river for the limit, southern limit of this Colonia Dignidad. So all the information and the works that we have performed have to do with the information provided by the witnesses, whether to the judge in charge of the investigation, the several judges in charge of this investigation, and also to us, the team of archaeologists who have been working and participating with the witnesses. For instance, in the case of the illegal Antigon in 1978, this is the information that we have by the witnesses. Obviously, I'm not going to mention who they are because this is judicial secrecy, but this is the information of what they have said. And this is exactly what they say. One of the witnesses with whom we discussed, they said the holes that we made was for digging the victims who were protesting against the military regime. The dimension of the holes were 2 meters deep, and the width is about 80 centimeters. They say these holes were made on the roads. I'm not sure how many holes were made, and he participated in 8 or 9 of these holes. And this was done during the night. After that, 5 years later, and according to this Retiro de Televisores operation, when the victims were buried illegally in different areas, there was an order from the central command, and they sent a sort of hidden message saying that they have to remove the TVs. And one of the witnesses, they have to unbury that with an excavator, with a machine. And the excavations lasted about 4 weeks. After that, they put the bodies in the buckets of the equipment. They removed most of the corpses, and they cleaned manually all the area with the rakes. After removing the bodies, they burned the bodies. And as we said, in Chile, the military, according to the information, they launched the bodies to the sea. But in this case, in Colonia Dignidad, they burned the bodies. The same witnesses. This is information that is included in the investigation. The bodies were put into bags and they were burned. According to one of the witnesses, they are about 20 to 30 bodies. And in order to make sure the bodies were burnt, they used some grids for construction. They put the bodies and they were bathed with fuel, coal, and also napalm. And the burn took about 3 or 4 days. So at the beginning, they burn according to the witnesses. That is what they say in the folder. They say to us in the interview that we had that the bodies were not burned as quickly as they wanted to. So they added this fuel such diesel, napalm, and also other components. The witnesses also said that after this, they took the ashes with the spades and they launched them into the river, Coloquio. With that information, that is in the judicial investigation and with the interviews that we made with the participants, we had an analysis of the investigation and a process implying each of these stages. Reading the process, interviews to the witnesses, analysis of the aerial pictures, archaeological inspections, excavations, reports to the relatives, and reports to the judge. The works have been done on and off since 2005 till 2018. We have excavated in more than 30 areas in the Andean area of Colonia Dignidad, and all these areas have been informed by the witnesses. We have also excavated in other areas that are compatible with the areas informed by the witnesses. They tell us that they buried the victims close to the road on the slope of a hill. We have found and searched for those areas. The general area is about 100 hectares, and this area is located far from the urban area. We're talking about 5 or 10 kilometers away from the Colonia Dignidad. With the pictures that you can see on the left, we can see a road, and this is one of the places that we identified in 2005. We did some excavations, and this is the road and the slope of the hill with a lot of vegetation. We're talking about the south. There's a lot of vegetation. And they said that on the road, they did all the holes. And also, they told us, and we made some holes here, some ditches, as you can see, in this area of the forest. But from the archaeological point of view, we found the following problem, that we have excavated some places that have to do with not the undigging, not the digging, but the undigging, because they made an excavation for digging and for undigging. So, as the witnesses says, the excavation for digging was very reduced, 2 meters deep, 2 meters long, 80 centimeters wide. 5 years later, they removed the bodies and the excavation they did is much bigger. So from the archaeological point of view, technical point of view, we have excavated some places in search of the inhumation area and the burial. And here on the left, we can see marks of the teeth of the excavator. It is not all the people participating in the same in all the activities. Some making the holes, and some others undigging the bodies. So the information is not shared by everyone. Here we can see some people of Colonia Dignidad where they told us some places where they could find the bodies. In 2017 and 18, we received information from witnesses, and our objective was to find the place where they burned the bodies. We were told a spot of about 1 hectare, about 10,000 square meters. We made some ditches, some trenches, in order to detect differences in the soil. And on the picture to the right, we can see it in January 2018, we found a clear disruption between the natural soil and this burned soil. Before this, we cleaned the area. We cut all the bushes and the trees. We widened the place and we started the archaeological excavations. On the left, we can see one of the witnesses. He is telling us the place where he says the bodies were burnt. You can see the differences in both places on the left and the right, where we had to clean the area in order to do the horizontal excavations. So with this methodology, we started the excavation in this area. And certainly we had the rains in winter, and in May 2018, we've completed the excavations of these areas where we could determine that actually this is an area burned. And here we can see some pieces of wood burned. And as you can see here, this is very shallow. And that is not the original surface, because when they burnt and removed the ashes, they do some raking to remove the ashes. So these are the works performed, and therefore our works in Colonia Dignidad have two senses: to find the area of digging and all the burning area. All the information has been provided by the witnesses, and that is part of the information of the judicial investigation. Of all the ditches, between 8 to 10, according to some of the witnesses, we have found only one of these ditches. The area, the burning area, that is how we call it, that was excavated in 2018, is compatible with a burning area, but we didn't find any piece of bone, only burnt soil. So therefore, in that area, we have proposed to make some chemical tests of the soil with labs from Switzerland and Germany in order to see if we can find with these sediments, with the analysis of these sediments, some components that the people added for burning the bodies. We know it's difficult because we're talking about 40 to 50 years after this, but this is a work that we have to do. These are the works that we have done, and our perspective and our balance is that the delivery of information of the witnesses has been incomplete, and therefore it is necessary that the participants, whether in the undigging of the exhumation, that are very few, provide the correct information for the location of the ditches. We, as archaeologists, need to do the excavations in the right places. If we don't have the information, we could have excavation in 100 hectares and with no results. even if we have the ditches removed, we have to find them and to do the right excavations in order to find bones, in order to have a DNA test, in order to identify the victims. We, for instance, in 2001, in the ditch that we found where the bodies were removed, this is part of the people who were killed in the assault to La Moneda. There were some bones, and through the analysis of the DNA, we could identify more than 11 people with fragments of bones. And that is what we want to find in this area, in these holes where we could find the victims, even if the ditches were removed, but we can find some sediments with fragments of bones or to make analysis of the DNA. On the other hand, the chemical analysis of the sediments could help us to define if this is the original burn or it is just a forest burn, which is very usual in the south. In the place, it is possible to continue with the excavations, but we have to be certain about the places. So the work to be done is very much. That is my presentation. Thank you very much, Ivan, for sharing your presentation. I think that is a good base for the analysis and the context that you're facing. What are the challenges, scientific challenges that we have for the future? We will continue with Francisco Echeverría.
Francisco Echeverría es doctor en medicina por la Universidad del País Vasco y también antropólogo forense. Es miembro de la Sociedad Científica Aranzadi desde 1973, del cual ha sido presidente.
He's presided, and is a scientific Spanish scientist that has been working since 2000 in the conflicts in Spain and the Civil War. He has also participated with many colleagues within the Falkland Islands, Malvina Islands, my apologies, and also in Africa. So Paco, as we call him, welcome. You have 15 minutes.
Muy bien, ¿se me escucha por ahí? Saludos.
Hello, can you hear me?
Muy bien, pues encantado de estar con vosotros, ya que
Pleased
yo
to
me
be
siento
with you
entrañablemente
since
unido
I feel very much
a vosotros como técnicos,
obliged to
especialistas y
you,
particularmente
specialists,
a las gentes
technicians,
de Chile,
and
por
also
supuesto que
people
tienen
from Chile
también la
that
pequeña oportunidad de
that
intervenir
they
en
also
muchos
have an
foros
opportunity
que
to
me
have
permiten
had
enriquecerme
chances
en
to
conocimientos y sobre todo
improve my
mejores
knowledge
ideas.
and
La
also
tragedia
the
civil de Chile,
tragedy, feel the
particularmente
tragedy
el 23,
on a human level.
me lleva a
Tragedy of
hacer el siguiente
1973
comentario.
makes me
Cuando
say
en
the
España
following. When
finalizamos una
Spain had
dictadura,
finalized a dictatorship
aproximadamente en el año
about
75-78, en realidad
1975,
ustedes
78,
estaban
you
inaugurando
were
su propia
inaugurating
dictadura,
your own
que es una
dictatorship,
cuestión
which is something
verdaderamente injusta.
unfair, unjust.
¿Ven la pantalla en este momento?
Can you see the screen right now?
¿Ven la presentación?
Can you see that? Yes, we can.
¿La ven ahí?
Perfecto.
Vale,
Yes.
muy bien. Bueno, pues esta sería mi presentación.
Well, this will be my presentation. I just want to make a comment. If the crime investigation is still in effect, well, it has been for 50 years. We are attempting for France that takes us to 80 years in a takeoff. It seems far in the past, but it's quite present that we have a pending investigation yet for these crimes. We cannot invoke laws or that there's respectability of the law. Feelings do not prescribe or expire, and the right to know the truth has no expiration date. So I participated in several aspects with Pablo Neruda, one of the cases, and I want to state that the importance that it was that to have a mandate, to have observation, and also to when human rights were inaugurated in 2010. But before that, we had been collaborating with the medical service from this declaration. This is very important from 2007. that we've constructed a system to identify the victims that is sure for the families and for society. We've brought the best national and international foreign experts for the painful identification of the remains. This isn't done by journalists or an individual. This is done by the uh, government, the president of the government. Why is this so? Well, at this time, there was a problem. We don't want to go into detail, but it was a Patio 29 when we produced a series of errors in the identification. We have to understand that there are mistakes in many laboratories, but the important is to measure and understand the error factor, introduce um, a, a error factor just as in industries or innovation, the incidents and the accidents, as well as errors, um, happen sometimes. We have to be aware of that, not to uncover these, and we have to continue. So we created an assessment with the next panel of experts. I was invited to participate in this audit, and I had the opportunity to understand the entire universe of. Uh, so these initiatives also brought on to reinforce our medical our services, to have legal medical service, having new professionals, incorporating new labs, younger people. To have a final point, which I believe is that in Chile today, the, the legal medical service has the human means and a material means to be able to carry out these tasks in the proper manner. Now, the key aspect of the process was that Michelle Bachelet, which is the president at that moment, Dr. Maria Luisa Sepulveda, which many of you know, and also with the full awareness of this firm decision to uh, be involved. That she's the one who referenced us, and how we qualified within the Chilean administration. People who still, from that moment, from the church, the victory of solidarity, who was had information and who had information which has key in findings and is in legal.
Pablo Neruda, lo he dicho antes, Víctor Jara, el presidente constitucional Salvador Allende y etcétera, muchos más. ¿No? Bueno, aquí tenemos esta fotografía que la quiero destacar porque sin ella no hubiera sido posible. Y veremos con mi última diapositiva.
So we see that I'm going to continue with the slide. Sorry, there was an interference. Some come from the when I go to foreign countries, I try to learn also. I'm not giving lessons. They are not the same in one place and the other. And we have to accommodate ourselves. We have to be able to recognize the capabilities that this is like in any country. Let's not think that every time we come into a country from outside that we're being invasive. We understand the legitimate practices in forensic within each country. So we can formulate documents what everyone else knew. The chain of errors has been maybe had progress because of maybe lack of means or skills or competencies that the professional had, and also lack of sufficient professionals in a with worked out with them without protocols or standards, the proper standards for the times that we were carrying these out. Fortunately, these changes that were being introduced into the uh, medical service that produced a boost of my very loved by everyone else, as you must know, not being a forensic physician had the conviction and the mandate on an institutional level to bring these tasks forth in a proper manner. It was a satisfaction for us because I will always say that Chile put at my disposition for our team all the material means that we would have as we started going down the path. Here we see the image where we have Shabbatale, Maria Sepulveda, and the book that we made is to be a forensic science wall of fame of Chile. Here we see the same members of the team that we were giving, providing the last report. I'm not going to free to the food. But we talked about a need to introduce improvements.
propio laboratorio de ADN del Servicio Médico Legal, que finalmente va a tener que ser la institución que
And
resuelva
for
estos
me,
análisis con independencia de que
that
pueda
even
estar
though
al
I may
100%
result in out,
de
we
las
need to have the
colaboraciones que
independence
llegan de cualquier otro
of the
lugar del mundo, pero
collaboration
tienen que ser ustedes,
from anywhere in
vosotros,
the
en
world.
el
But they
país
have to be you.
esos profesionales
You
los
may
que tengan que
you may
resolver
allow me to say
que se avecinado
the
un poco hacia el futuro.
have to look
Reconocemos
at
los
have to
errores,
look
de
toward the
acuerdo,
future
no
as
se
well.
trata
We
ahora de
recognize
descalificar a nadie
errors.
de quienes con buena
Obviously,
voluntad intervienen
the
aquí
idea
hace
is to
tiempo, pero que realmente
qualify
fueron
the
demasiados
good
errores,
willingness wanted to carry this out, but there were too many
muchos,
mistakes,
muchísimos. Y claro, como
many
la foto
mistakes.
es más completa, aparece la razón del
Obviously,
rey
this
también,
is the
para
image that's
señalar
more
que estos
complete
procesos,
uh,
cuando son así
image of everyone
tristes en su
participating. Now, these
conjunto, pero a su vez
processes,
como un
when they're
problema
so sad
institucional,
and
porque
at
el
the
servicio
same
médico
time as
legal,
a problem,
si
it's an
se querrá
institutional problem because the
descalificarlo, pues tienen que
medical
salir de esa
forensic
situación
service,
y entonces para
if
eso
it was
necesitan tiempo y
qualified, they
medios.
had to be
Pero
taken out, and
en estos procesos
there are no
suele
means
ser
at this
habitual que existen
process. So
observadores
usually there
internacionales.
are
No es lo
international
mismo los
observers.
observadores
It's not
internacionales que los peritos
international observers
internacionales.
or
Pero es
legal
bueno y se ha hecho
forensic.
efectivamente
They're not
en
the
Chile
same.
que con
Again,
independencia del
that
equipo
time
policial
in Chile,
existan también
usually
otros observadores
there are other
que ven el
observers
proceso,
that
que lo
maybe I
critican,
should have been
que lo
included,
corrigen
criticize it,
sobre la marcha
observe it,
y que
and
dan algunas
allow
garantías,
correction,
digamos, a la sociedad
and that will give
chilena y también a la
guarantees to
comunidad
the Chilean
internacional.
society and also
Así
the
entonces se
community.
creó además el
The
Comité Internacional de
so
Expertos.
how
Él fue el que
there's
puso
an
algunas de las
international
condiciones
expert
para ver
committee
hasta qué
which
punto
placed some
los análisis
conditions
que se querían
to
hacer,
see up
los
to what
sofisticados
point
de
the
laboratorios
analysis
genéticos y demás cumplían unos
sophisticated
estándares
genetic
que para Chile se
laboratory
pusieron muy altos,
or
muy
standards
altos,
that
incluso
were
nosotros
very high,
buscando, aunque yo no
higher
conozco
than
todos los
other
países
places
como os
in the
podéis
world.
imaginar, al menos sí se procuró
As you can
que los
imagine,
estándares
many
fueran los
countries,
mejores
but
posibles.
here the
Este
standards
Comité
are the best.
Internacional de Expertos
So
contó
this
con personas
international
además
committee of
cualificadas
experts,
en
the
materia de
most
identificación
qualified
humana, muy
staff
particularmente en
within
materia
matters
de
of
genética forense.
uh,
Estas son algunas de las
forensic
fotografías
genetic
en
examination,
las
because
que es
some of the
importante
images
destacar,
where
haremos a
it's
partir de
very
agosto,
important
esto
to
lo dije en abril,
highlight
pero están los
this,
familiares.
this, this,
Los
this
familiares
direction.
están
There's
aquí,
a
no
family.
están
So
alejados.
here,
Esta
we're not
experiencia
far
de
away.
Chile
The
pues,
family,
se
us
puede
to
trasladar a
tell you
cualquier
the
lugar del
experience.
mundo.
We could bring it
Cualquier
anywhere in the
proceso
world for any
de
process,
análisis, de estudio
the
forense
analysis of
en
forensic
materia de
studies
vulneraciones de los derechos
in
humanos
terms of
debe contar con la
human rights
proximidad,
vulneration
con la
should have the
información, con la
proximity and the
representación de las
inclusion
familias. Las
and the
familias tienen el derecho
representations
a
of
saber
families. They
no
have
solo
the
qué está
right to
ocurriendo.
know
Las familias pueden no
what is
saber de
happening.
ADN, no
Families
importa,
may
pero
not
sí tienen derecho a
know
hacer un
specific details
seguimiento
on
cercano. No
DNA, but
pueden estar
they
lejos.
have to be able
Lo
to
digo porque
follow
yo
through.
como
They
médico
shouldn't be
forense
far.
pues
And I
me he
say this as
manejado
a
muchas
forensic
veces en esa frialdad que
I've
tiene el ser
worked many
funcionario
times
público,
in being
casi
in the
no poder
cold
hablar con las
environment
familias,
of
el estar
being in a public
sujeto a la
role and
autoridad
not being able to
judicial,
talk to
el
the
rendir
families
cuentas,
and not being able to
eh,
being subject to
el
own
rendir
legal
cuentas para los
positions
jueces,
or
fiscales y
provision
tribunales
to give
y no poder
accounts to
hacer un
court
acercamiento
and
a las
judges,
familias. A
but
las
not
familias
be able to
les resulta
to the
relevante,
families.
importante conocer
It's
a los peritos,
relevant to
a
the
los
families
peritos, conocerlos de
as
cerca,
well is to be
conocer
just to
en lo
understand
personal, incluso nuestras propias inquietudes.
personal
Eso
aspects
es
maybe
útil porque tienen que
that's
estar
very
insertados
useful
en este proceso.
because they have to
Quizás
be
yo he visto en
inserted
Chile
in this
estas
process.
cosas
Maybe I've
relativamente
seen
sencillas,
Kelly
¿no?
these
Hay una
things.
voluntad política,
They're
hay una
simple. There's a
cooperación
political willpower. There's an
intersectorial, hay un
interterritorial
sistema judicial
cooperation.
que
The
afortunadamente
legal
está
system
implicado,
that
los
is
procesos estaban
implied,
judicializados, pacto 29 y los
judicialized,
demás, y hay unos activos
and there's
forenses que, bueno,
also
se pueden
a forensic
mejorar,
process
consolidar,
that
reforzar
can
y
be
renovar.
consolidated,
Ahora,
improved.
¿qué es lo que ocurre? Lo
So
estaba
what
explicando el
happens
doctor
then,
Iván Cáceres, claro.
as the Dr.
Es que
Ivan
no
Cáceres was
solamente
saying,
les mataron,
um,
los
it's
asesinaron,
not
sino
that
que los hicieron
they would be
desaparecer
killed.
con la intención de que
They
no
would
los
disappear
encontráramos
with the
nunca más.
intention that no one
Nosotros en
find them
España
ever.
tenemos
Now in Spain,
aproximadamente
we
unos
have
114.000
approximately about
desaparecidos y solo
114,000
hemos encontrado a
disappeared,
9.000.
and we've only found 9,000.
9.000 puede parecer mucho, pero en 20 años solo
9,000
hemos
may seem a lot,
encontrado
but in
9.000.
20 years, we've
¿Dónde están
only
los otros
found
100.000? Es que no los vamos a
90,000.
encontrar,
90,000.
seguramente
So where are
nunca,
the other
porque esto
remaining
tiene limitaciones.
100,000?
No vamos a poder
No,
encontrar
it's limited. We're
todas
not
las
going to
cosas,
find them
ni vamos a
all.
poder identificar a
And
todos los
it's just
supervivientes, quizás en
we're not
algunas
going to be
zonas,
able to just
porque
cover
en tiempo
everyone
de
because
la guerra y
time
posteriormente, pues en
war
España se
popular.
recogieron
Well,
perseguidas por los acantilados
many,
del mar.
many people were
Esto
thrown
no requiere
into the
aquí
sea.
una
That
técnica
doesn't
arqueológica
require
ni
any
nada, eso es
archaeological,
completamente
uh,
imposible.
that's
Y es
totally
cierto que vosotros
impossible.
conocéis bien
So what we
llevar a
know,
estos
we take
militares. En el año
the Spring
73
Festival
se
Comet. In
cumplieron 3 actos
1973,
clandestinos. La
there were
Asamblea
also
Naturaleza sí,
um,
sí
the
intervino en la
scientific
vuelta de los cuerpos,
legal
pero no hizo las cosas debidamente. Los informes
experience shows
militares
that
valen
they
una
were
decisión
able
muy poco.
to
Tenemos por
intervene
ahí
and
el
find
informe
these
de causa de muerte
illegal
de la
burials,
que sí fue
but not everyone. We see Victor Prada was identified as Victor Prada. Well, that's Christian of the report. Apropos uh, so many aspects that need to be looked at. Those who sublevated, and from 1974, there were up to maybe 19, it was the most difficult uh, period to find we find these bodies without hands or they were deformed. Maybe hands were that was a way that was a way of not finding the the any identification. But as you know, in 1978, there was a massive unearthing um, that was hitting the television extraction, which is very humiliating. The name that was given, the operation given to uh, responsible public officers that have certain responsibilities, name an operation as such involving remains. So there uh, I guess, unfortunate conditions, those who are affected by these circumstances was one of these examples only. It's where they practice an operation to retrieve televisors. I'm not going to entertain you with this layout. The effort they took continue with qualified personnel to prepare and to um, burn, to incinerate. If they knew how to do it, it wasn't just any just someone who's supposed to burn a body, and they were trying to, they want to burn them up to incineration, as we see with some cases. So it's easy sometimes that someone can understand that we find the full body, that it's not going to be the case. And for Colonia Dignidad, from what has been saying, in the case of kids that have been removed or we have found some remains and small bones. Uh, Even with this archaeological technique, sometimes it could be maybe a small fragment, a distal bone, a rib, but it is not detailed. Well, it's fine. That happened to them too. And so many of these have been able to give positive identification. Now, the economics, even in the characteristics in the removal bones that have been left out in the weathering, and things are more difficult. Here are the small flagged out bones that cartilage like that has been able to be found in some places, the cartilage, even after the reviewing of the television set operation. Also, we find these to take a maximize the finding to identify making that positive finding, even when we have this fragmentation. So if this is burned, then what if the skeleton is burned? This is a complete case of Chile, where it's all the bones are also burned, and then carbonized, it's not information. So there's parts of the organism that have been totally destroyed. There's a lot of bones, even teeth. And what would be possible then, if they genetic analysis to show how these are those more ventilated or those are burned, basically ears burned, and knees, the most exposed to this stuff. It persists with the burning, we have a total incineration. So with all of these conditions, it has been possible to find an identification, even if it burns, as we could say, carbon, but not integrated. These are some of the examples of what we identified in the forensic medical service. Yeah. So this is another example where we see that the body has been carbonized. We see we see the sound, but from these, it would be totally possible to extract DNA. Sorry, every we are able to make improvements in this in finding the hard elements. But we theory uh,
Este es un
seeing
caso
this
que
that
parece
when
mentira,
something,
publicado por el
this was a case,
doctor Ángel
a
Carracedo y
Dr.
miembros de su equipo,
Carracedo
que es
and
un
members of
penalista
his
que ha
team,
colaborado también
he was
con la Asociación Médico
collaborating
Legal de
with
Chile.
the
Es un
Chile
penalista
medical
importante en España, su laboratorio en la Universidad de Santiago de
Clinical
Compostela está formando
laboratory
muchos
and
profesionales.
Chile de
Y
Compostela. Many professionals
es
have
un
been
ejemplo de un cuerpo
as
que
an
se quemó
example of
como
body
consecuencia de un incendio
burn
forestal.
of a
El
forest
cuerpo pues
fire.
está entre
The
incinerado
body
y carbonizado,
is being
pero no
carbonized,
obstante había
hard, and
porciones, algunas partes
incinerated.
de
But there was
ese
some
hueso, en el
portions
que en
of
general es
any part of the
imposible
bone which
obtener
is almost
ADN, alguna
impossible to
parte
get
en
it
la que
made
sí fue
to
posible
where,
obtener
and
ADN,
some of
y
the
de
pieces
hecho fue
where it was
identificado.
possible to get
Se publicó como una
DNA,
cosa
and it was published
realmente
as a
difícil porque la
difficult
temperatura de
condition
carbonización
because
suele
the
ser en torno a 300 grados
carbonization
o
to
más.
300
La
degrees
investigación requiere unos
and
600. Pues muy bien, un
generation
cuerpo que
requires
haya estado
600.
a
So
300 grados todavía podría tener
300
algunas
degree
porciones en
exposure
donde el
may
hueso
have
propiamente
some,
esté indemne.
um,
Aquí vemos
again,
hueso
loose
completamente incinerado,
won't
sería imposible
totally
obtener
separated.
ADN.
It would be
Aquí vemos
difficult to
unos
get
huesos que están
DNA.
un
There's
poco
some
más
bones that are a bit
conservados y aquí sería perfectamente
better
posible.
conserved
Bien,
where we could
entonces
find
lo siguiente que voy a
NFX.
decir, y voy a
So
finalizar mi
find,
exposición, que
I'm going
no
to
he dicho
finalize
nada
my
nuevo,
presentation,
en
which
realidad
I've
esto lo
not
tenía
said
pensado
anything
porque
new.
Cáceres ya lo ha
This
explicado
was,
además de ser
Ivan Cáceres, I
oyente
think,
en su
had
tesis
explained this
doctoral, la
in
Iván
his
Cáceres habla
council
de estas
thesis. As
cuestiones
he
y yo con toda la información
all the information
realmente valiosa
had
que ha aportado Iván
valuable that
y su
Ivan has
equipo
provided
y la
in his team
investigación judicial, me
and
pregunto lo
legal
siguiente, y voy a
investigation.
concluir
I
ya,
want to
¿no?
know.
Se ha generado prueba,
There was
se ha generado
evidence,
evidencia, no
evidence,
arqueológica, pero sí hay
no
evidencia,
archaeological,
hay
but there's
evidencia
evidence now.
forense.
Now we have
No hay,
forensic
me parece que la
evidence.
ciencia forense no
The
es
science,
solamente el
forensic science is
hueso
not
y
just
el
the bone
antropólogo o
and
médico forense, es
anthropologist
más,
with
aquí hay
medical forensic
tarea para químicos
legal
forenses y para ingenieros
processes,
forenses y lo que
chemicals
queramos,
and
¿no?
forensic
He
engineers.
sacado incluso información
That
de algunos
I've
vídeos.
extracted some
No vamos
information
a
from videos. We're
pararnos
not
ahora, pero
going
no tiene
to
desperdicio
can't
cuando en
stop on
estos
these, but
noticiarios
when these
aparecen autoridades
give
en
the
el lugar,
information to authorities
aparecen
in the place,
imágenes,
and we
aquí
see
está Iván
images.
hablando de una
Here,
de
this
las
is the
fosas,
land and
y
one of
aparece
the
incluso el señor que condujo la
the
máquina,
people who drove the
es el
machine,
que exhumó los restos. O sea,
the
y entonces yo
one
me
who
pregunto, y
elected
finalizo
and
ya, Luis,
conducted.
perdóname.
So I'm
Vamos
still
a ver,
not this. So
la constitución de la prueba en
we
el
have
ámbito forense
um,
es mediante
witnesses
la
and
información
evidence. So
documental,
how do we
la
manage
información testifical
evidence?
y la
Yes,
información
documents,
pericial.
the testimonies, and
Nosotros,
the
los
legal
forenses,
forensic.
estamos aquí en lo pericial,
Okay, this is where
la
we're at
magia de la
now.
ciencia
The
forense, ver lo que
magic of
sale
forensic science,
en los
what we
operativos y
see
en las
in the
televisiones, pero un
television.
momento,
But
pero no hay prueba
wait a minute,
documental y
but there's
testifical
no
suficiente, me pregunto
documental
yo. Es
and
más, la prueba pericial no
enough
ha aportado
witness
información útil en cuanto a la
testimonial of
exhumación. Yo diría que
this
sí.
is the
En
papers
cuanto a la
of the
exhumación,
legal
hemos
forensic
encontrado
is okay.
el
The
lugar de los hechos.
burying
Claro que no se ha
has
identificado
been
ninguno y no se va a
has,
poder establecer la causa de la muerte.
have
Pero
not
nosotros
found
estamos todo el tiempo
specific
transformando
documents that,
indicios
but
en
we are
evidencias.
transforming
El
in
juego forense consiste en
small
transformar aquel indicio en una
pieces into
evidencia. Y si
evidence,
se
and we need
valida,
to transform, and if it's
la
validated, such
validación no lo hacen los forenses, ni los
validation
congresos de los
it's not done
forenses,
by the
ni el servicio médico
forensic
legal. Son
assistant or
otras
medical
autoridades,
legal
son las
the
judiciales
authorities,
las que pueden
legal
convertir la evidencia
that
en
can
prueba.
convert
Y entonces finalizo
evidence
con esta
into
imagen, que me parece
evidence,
relevante para
into
entenderlo.
the
¿Y
findings
por
and
qué no la
evidence. So
aplicamos más?
why
Es
don't
que
we
el
apply this?
operativo
Well,
forense
the forensic
parece que esto solo de
investigation, which is
expertos en
this is
bioquímica, en
from
genética,
only
en
genetic and
biomateriales.
experts in
Pero
bio.
no, un momento, aquí cada
No,
uno tiene
no,
lo suyo.
we
El
everyone has
principio
a
de
role.
legitimidad,
The first
de
of the
legalidad, de seguridad,
legitimacy
de
and
recursos
security
y de
and
gerencia y
resources
consolidación de la
and
información, esta pirámide tiene
of
que
this
funcionar
information
bien.
has
Para
to
que funcione el
operate
operativo forense
so
tiene que
that
estar
the
bien
forensic
estructurado todo de ahí.
operation is
Y
well
todo de ahí abajo
structured
no es
and
cuestión de
everything
forenses,
below.
ni de
It's not
bioquímicos,
just
ni de geneticistas.
biochemistry
Es
or
cuestión de
genetic
otros
experts,
profesionales. Y
other
de otras autoridades.
professionals,
Se llama así, esto lo que
other
tiene que tener
authorities.
es
Call
credibilidad.
these
Muy bien.
call this
Entonces llego
pyramid
a
a
mis conclusiones.
credibility. So
A mis
my
conclusiones.
conclusion,
Yo creo que en Chile hay medios
I
humanos y
think in Chile
materiales
they are
suficientes
human and
para
material
investigar el caso de Colonia Dignidad. Con
colony.
todo, esta
For
investigación debe avanzar
this
en los
investigation
aspectos de
should
análisis policial y
progress
análisis judicial.
and
Porque es que
legal and
igual, es
police
triste decirlo, pero es que igual no
analysis
se
because
encuentra nunca una
we
evidencia
never,
arqueológica. Y si
we
está
will not
además muy quemada,
find
no va a
full.
rendir para obtener
It's not
ADN.
going to be enough
Esto es una
for one
tragedia para los
community.
familiares.
This is a
Decirlo es muy
tragedy.
duro.
Say it's
Decirlo
very hard to
de
say
la
it
manera en que yo lo estoy
in the
expresando.
way
Pero
that I'm
hay que hacernos
expressing it.
a la
But
realidad de que
we have to face reality that it will not be possible just on that. We have to give it an input through a commission, experts, so on, to review everything, to look at the entirety of the information, not just what Ivan has explained, but everything that has been found. Or isn't it enough for a forensic knowing that the crane driver, oh, the bodies are right, they were then they burned them and used it. I mean, his testimony wasn't that also added some finalizing that. So that forensic science cannot do everything. It has some limitations. And like in this case, at least up until now, like unfortunately, military did it so well, they were able to even have all evidence being launched or thrown into a river. Maybe this and maybe a tragedy for the families, but the families have the right to know the truth. They have the right to justice, and it's the legal authorities that we have to intensify the investigation in other directions and to understand that the forensic society in Chile as well as the rest of the countries are in position to support everything that results, especially in the most scientific, technical aspect of the issue. Thank you.
Gracias, Paco, por la
Thank you,
presentación. Creo
Paco, for the
que hemos puesto en
presentation.
contexto más amplio los trabajos
Um,
que había explicado Iván y hemos
this is some context to
escuchado
the
la
information
experiencia
provided by
que ya tiene
Ivan.
Chile desde el
Um,
patio 29 en adelante,
Patio
los
29
testimonios
has
familiares
were given
y
results and
creo
what
que
expectations
estamos
families have.
avanzando
I think
bien
we're
en este sentido.
progressing in this sense.
La próxima
Now, the following
presentación
presentation is
Jennifer de Brum,
Jennifer
es
de Bruin,
microbióloga,
she's a
especialista en
microbiologist,
medio ambiente,
specialist
es
in
profesora
uh,
asociada al Departamento
an
de
environment.
Sistemas y de
She's
Suelos en la
a
Universidad de Tennessee, en
member of
Knoxville,
an
Tennessee,
Knoxville,
y
Tennessee.
su área de trabajo
Yeah,
tiene que ver
her
con la
area of
actividad
expertise
de
with
microbios en el
micro,
proceso de descomposición.
microbiology
Trabaja
decomposition
hace más
work
de 10 años
in over
en la
10 years
famosa
in
granja de
famous
Calabres en Tennessee. Así que
Tennessee
Jennifer, bienvenida y tenés tus
laboratory.
20 minutos.
We have 20
Muchas
minutes
gracias.
for you, if you could please.
Thank you. Can you all see my screen? Can someone answer? Does someone see my screen? Are we good?
Yes.
Okay, thank you. All right. Well, thank you very much for having me. It is my pleasure to be here and contribute to this very important discussion that we're having here this afternoon. In my talk today, I will be discussing some of our current research into signatures of decomposition in soil systems. Before I get into signatures of soil that are relevant in this case, I wanted to give you a very brief introduction of some of the research that I do in my lab and some of my expertise. A lot of the research we do is focusing on decomposition of animal carcasses in natural ecosystems, looking at nutrient flow and recycling. We also do research on human decomposition, and a lot of the work we do related to human remains decomposition is in collaboration with the University of Tennessee Forensic Anthropology Center. The Forensic Anthropology Center runs the Anthropology Research Facility. This is also known as the Body Farm. It is a 3-acre outdoor laboratory that is dedicated to studies of human decomposition. It's a very special and important place to work because it allows us to conduct longitudinal or long-term studies of decomposition. And much of the research done there is inspired by actual forensic cases. The facility has been running since 1980, so almost 40 years now, which has given us a lot of really good information about human decomposition. And the success of the facility is largely because of its large body donation program. Approximately 100 donors per year are decomposed at this facility, and we have about 4,000 or more living individuals that have pre-registered to donate their body to the facility after death. So it's a very important and special place to work when it comes to understanding processes of decomposition. And then in our work, what we do, both in terms of animal and human, is focusing on microbial and soil responses to decomposition. The questions that I'm going to be focusing on here today include the... Sorry, did someone say something? No, okay. The questions I'm going to be focusing on here today are the changes that we see in the local environment as a result of decomposition, and what signals are most likely to persist for long periods of time. So in the case of Colonia Dignidad, we're talking about perhaps 40 years have passed. Which of the signals of short-term decomposition might we expect to persist for a long time in the soil? So decomposition of animals or humans impacts the local soil environment pretty significantly. And we call these areas hotspots, decomposition hotspots. When animals or humans start decomposing, it actually begins internally. The microbes, particularly that live in our gut and in our lungs and on our body, they start by doing the initial decomposition. This liquefies tissues, and the liquefied tissues and decomposition products are then leached into the surrounding soil environment. All of the active microbes and all the nutrient-rich fluids that are coming out of these decomposing bodies then stimulate a lot of local activity in the soil. So microbes that are already in the soil receive these decomposition products, these nutrients, and it causes their activity to increase. So what we end up is in something that we call in ecology a hotspot, and that is a local area or a patch where we have enhanced microbial activity and biogeochemical cycling. So a lot of things turning over in the soil. And the other thing to note is that these hotspots are often ephemeral or temporary. After the resources, the nutrients are used up, the soil, we think, mostly returns back to normal. But the timing of that can vary widely from case to case. This is what one of the hotspots might look like. In this case, this was some research we did using beaver carcasses. This is a North American beaver. Shortly after death on the left panel, and then on the right, you can see after decomposition, there is actually visible discoloration of the soil where this hotspot has occurred. So what's going on in this hotspot? Well, we've spent a lot of time investigating some of the microbial activities and the transformations in the chemistry in these systems. This is kind of a complicated schematic.
Sorry, Jennifer, there is a problem with the translation.
Okay.
Hold on 1 second. Luis.
Se escucha traduciendo.
Yo escucho a Verónica.
¿Está traduciendo bien al español?
Yo también estoy escuchando a Verónica.
Ok,
No,
Jennifer,
es que Verónica la estamos escuchando perfectamente.
No, la gente que está en Facebook me están diciendo, en Facebook y en YouTube, si la están escuchando.
They're just confirming the Facebook interpretation, but they've been hearing me fine.
La traducción no se está realizando en Facebook, es un error del sistema, no sabemos por qué, lo estamos solucionando.
There's a mistake for Facebook, but
Go ahead, Jennifer, sorry.
Okay, thank you. So in these hotspots, this is a diagram that shows some of the major changes that we see in the soils during decomposition of animal carcasses. And while it is a little bit of a complicated figure, I'll draw your attention to some of the key changes. So one of the things that we see is that the decomposition, the microbial activity, uses up a lot of oxygen and tends to make the soils go anoxic. It actually draws all of the oxygen out of the area. And so we see during decomposition a period of anoxia or anaerobic soils, and that's colored in gray in these two panels. The other thing that we see in soil regularly is a pulse of ammonium. So ammonium, or NH4+, is a breakdown product, particularly of proteins, so muscle tissues. And that ammonium is in very high concentration in decomposition fluid, which then enters the soil. And so we see this big amount of nitrogen in the form of ammonium. that ammonium tends to persist in the soil really until soil oxygen comes back. And that can take a varying amount of times depending on the local soil conditions and the environment. But what happens is once oxygen returns in the latter half of decay, that ammonium can be converted to nitrate. All of this cycling of nitrogen also leaves a mark on the nitrogen isotopes in the system. And in particular, looking at the heavy isotope of nitrogen, or N15. So we, animals and humans, our bodies are enriched in nitrogen 15 compared to the normal isotope, the lighter isotope, which is nitrogen 14. And so when bodies decompose, the higher amounts of 15N, along with a lot of the microbial activity that's converting those nitrogen products, increases the amount of 15N in soil. And we can actually measure that isotopic change. So here, in another experiment, we looked at the distribution of those isotopes, the change in those isotopes in relation to that hotspot, looking both laterally away from the hotspot center and horizontally down into the soil column. And the data that you see presented here shows the difference between a control nearby soil in dashed line and the hotspot soils in a black line. And so you can see that we do have enriched amounts of 15N associated with that hotspot. We also see that 15N enhanced moving away from the hotspot up to about 40 or 60 centimeters in the case of smaller animals like beavers. We didn't see any changes in the carbon isotopes that we measured, 13C. We also noted at this particular site, we've been able to detect these isotopic changes 2 years after decomposition. We've collected samples this year, which will represent 4 years of decomposition. So we're in the process of measuring to see if that signal has still persisted. We also conducted a mass grave or multi-individual burial experiment. This experiment was actually a large part of Alex Emmons' PhD dissertation project. You'll be hearing from Alex Emmons next. In this experiment, 3 human subjects were interred in a grave, and then 4 years later, we excavated and measured a lot of the soil parameters. The ones I want to share with you today are the ones that we saw mostly changed after 4 years of human decomposition. We saw evidence of nitrogen enrichment, so high ammonium and dissolved organic nitrogen, and elevated electrical conductivity, a measure of soil salts. We also noted that the isotopic signature was still changed in this grave too. We saw an enriched 15N at the top of the grave, although this was most likely due to the disturbance itself. When you bring deep soils up to the surface, that also changes the isotopic signature.
Sorry, Jennifer, still there is a problem with the translation. Luis, ¿se puede arreglar la traducción?
Jorge está por ahí. ¿Me escuchan?
¿Están ustedes probando?
Por favor, Diego, por favor, ¿puedes cambiar el canal a donde está Jorge para continuar, por favor, con la traducción? Porque él se escuchaba bastante bien.
No, es que ya, cambié al inglés. Donde el canal donde estaba Jorge.
¿Puede continuar?
Ya, cambié al inglés. Donde el canal donde estaba Jorge. ¿Puede continuar?
¿Está listo ya?
Sí, continuemos.
You can go, Jennifer. Sorry again.
Okay, no problem. So when we looked at the N15 or isotopic signatures from this mass grave experiment, we saw enrichment at the surface of the grave, but this was most likely due to disturbance from bringing deep soils up to the surface. That will cause an enrichment in signal. But we did see enriched signal at the bottom of the grave, which we think is due to human decomposition. So we were detecting enrichment below as well. The final part I wanted to mention is some of our work on the microbial communities that are at these sites. I'm a microbiologist by training, so of course I have to pay attention to the biology. These are really interesting systems when you start to think about the local microbes and how they might be affected. In particular, microbial communities in these areas are really a mixture of microbes that are originating from the carcass or from the human remains itself, all those decomposers inside us. It will be soil microbes that respond to the nutrients and are becoming quite active. We can also have microbes that are being introduced by other visitors to the remains, such as scavengers or insects that can access the remains. So we end up having a very unique and different environment for microbes and microfauna to exist. So we have been investigating some of these communities, and I just want to mention very briefly some of the things that we consistently see. We've looked quite a lot at soil bacteria in these hotspots, and the one thing that we consistently see is a shift towards anaerobic taxa, microbes that can tolerate low or no oxygen. That makes sense since this is something we We've also looked at nematodes, which comprise of a variety of trophic groups. So they have a lot of different lifestyles, but are very prevalent in soils everywhere. So we've looked at nematodes to see how they respond. And the one thing that we're seeing there consistently is that as decomposition progresses and these hotspots form, we see a bloom of bacterial feeding taxa. In this lower panel, it is in the red. These are nematodes that love to feed on bacteria. This makes sense as we see a stimulation of bacteria in the system, that those that like to feed on them would also be enhanced. So thinking about some of these signatures and whether they may persist long-term, you probably noticed that most of the work we're doing is relatively short-term, very interested in soft tissue decomposition. But for this panel, I wanted to think about which of these might be useful for long-term signatures. And really what we're seeing across multiple studies is that the most likely soil parameter to remain altered over a long term is this N15 signature that we're consistently picking up. So in our studies, we've looked at beavers decomposing on a surface soil. We see that after 2 years and maybe longer. In a saturated burial situation with human remains, we see this signature at least 4 years later. We are in the process of trying to figure out if we see it even longer than that. So we have at our, for our advantage, working at the body farm. I mentioned earlier, it's been in operation almost 40 years. So we're currently testing soils there to see what 30 or more years of decomposition has done, and if we're able to pick up the signature there. And it's also important to consider what controls the persistence of these signals. What's going to affect how long these signals would still be detectable in soil? And that really comes back to two of the main controls on decomposition rates itself. And the first of those is temperature. Under warmer conditions, microbial activity is higher, decomposition rates are going to go faster, and we expect those signatures to not last as long. Oxygen is the other key control on microbial decomposition. So where you have oxygen, decomposition is going to go faster, and those signals will go away faster. And so thinking about the site in terms of not only the local climate, but in terms of the soil oxygenation status is going to be very important here. So if you're dealing with unsaturated soils where there is lots of oxygen, things are less likely to persist. Whereas if you have saturated soils, low oxygen, some of these signals may be persisting longer. And of course, I would be remiss if I did not point out the major limitation of the parameters that I've been talking about today. And that is that none of the things I've talked about will distinguish between animal and human. They could be signatures of decomposition, but really there would be no way to tell if it was a large animal versus a human. And so that is really where DNA-based approaches come in. And so this is really my conclusion and my setup for our next speaker, Alex Emmons.
Gracias, Jennifer, por la presentación. Yo creo que es muy importante porque hay muy pocos.
Thank you, Jennifer, for the presentations, because there are few places in the world using this testing with corpses, with the body farm, and you are the oldest working, and the first one, actually. There have been a lot of studies, and I think this is one of the best places for this kind of research and asking these questions about Colonia Dignidad. And in this sense, thank you very much. Now we are going to continue with the last speaker. This is Dr. She's a physical anthropologist at the University of Colorado. Her area has to do with when the DNA is degraded, the microbial activities and the relationships in terms of the decomposition of the corpses and the timing of the death. Alex, thank you very much. You have 20 minutes to go from now on.
Thank you. Just gonna share my screen.
Sorry, Alex, they asked me to stop to fix the translation. Just 1 second.
No.
Luis,
can you all see that? Hopefully the right screen is up.
Yeah,
Sí, escucho bien.
¿Está bien la traducción?
Sí, Luis, el problema que tenemos,
No.
me indican de la Universidad Católica del Maule, que están a cargo de la transmisión, es que al parecer Facebook no
Alex,
está...
they are saying that they have some problems with the audio, but this is for the streaming. This is not for the interpretation, so don't worry.
Okay. Should I continue?
No, just wait a minute, please.
Okay.
Yeah, we should wait for a couple of minutes because they are trying to fix the problem with the streaming. The problem is that the interpretation is not done into Spanish through the streaming. The Zoom is okay, but not the streaming into YouTube and Facebook Live.
Okay.
Okay, so we can wait just for a couple of minutes, please.
¿Me avisan cuando ya lo han resuelto para saber si podemos continuar?
¿Están en eso?
Comencemos para probar, por favor.
Okay, we can start. Just to have a test, could you please start, Alex?
Okay. Hi, everyone. Thank you for the invitation. It's a pleasure to be here today. Just as a reintroduction of who I am, I'm a postdoctoral researcher at Colorado State University. My primary areas of research generally relate to postmortem microbial communities and skeletal DNA degradation. But I've also worked on projects related to the persistence of human DNA, as Dr. Jennifer DeBruyn mentioned. And today I'm really going to be discussing the potential for eDNA to be used as a tool in human identification.
Are we all good with the audio? Okay.
Sí, aquí lo estamos escuchando
So
perfectamente
what do
en
we know
Zoom.
about
El
eDNA?
problema que
Well,
persiste es
eDNA
en,
refers to
en
environmental
Facebook.
DNA,
Lo estamos
which is
solucionando.
DNA extracted from an environmental sample.
Alex,
This D-
sorry. Just wait a minute. This still has problems with the Facebook, with this streaming. Okay, thank you.
¿Podemos continuar?
Jorge,
Yes.
lo que podemos hacer entonces es eliminar
So
este
what
canal de
Diego
audio.
says is that we are going to delete the audio. You can speak low, and we are going to do a sort of voiceover. So your voice could be a little bit low, and my voice is going to be a little bit higher, so that we can use the same channel.
Okay, so you want me to talk softly?
Yes, that is the idea, to try to use the same channel, because that's a problem. We will try to see if we can. Let's try to continue. Okay, if we can do it.
Okay. So as I was saying, environmental DNA refers to, or eDNA refers to environmental DNA, which is DNA extracted from an environmental sample. This DNA can be intracellular or within a cell, or extracellular outside of a cell, and can originate from a variety of organisms, both microscopic
Que pueden
organisms
ser macroscópicos.
and larger macroscopic organisms.
Son organismos macroscópicos grandes y
Potential
fuentes potenciales
from mammals
que
or
provienen
other
de mamíferos,
include feces,
de
skin,
heces, de piel, de
urine,
cabello, de orina, de
blood,
saliva, de
or
sangre o de
products.
productos de la descomposición.
A lot of the research involving
Gran parte de la
eDNA
investigación tiene que ver con el ADN,
monitoring
en donde
biodiversity,
podemos hacer el monitoreo de la biodiversidad,
and typically this biodiversity is in aquatic
y esto
systems.
está
This,
en los sistemas acuáticos,
this includes research areas such as
en donde
ecosystem,
existen áreas de investigación del ecosistema.
the role of rare species and conservation,
El rol es buscar y conservar estos componentes.
other topics, particularly in microbial
Otros
systems,
temas, especialmente en los sistemas microbianos.
around ecosystem dynamics and
Tenemos
understanding.
un ecosistema dinámico para poder entender cómo funciona.
Perhaps more pertinent to the discussion today,
Quizás
eDNA,
lo que tiene que ver con la investigación de ahora es respecto al ADN,
eDNA has also become a tool to understand past environment,
que es una
and DNA.
herramienta que nos permite poder entender lo que ha sucedido en el ambiente a través de los años.
So as you can see in the figure
Entonces,
here,
como podemos ver acá,
which is from a recent review paper, the
tenemos un
number
paper de una
related to
investigación reciente
environmental DNA have increased
donde nos
substantially.
indica el tipo de ADN que podemos encontrar en distintos ambientes.
And as seen here in B, mammals
Aquí
and
podemos
other
ver
large
en B
are not really contributing significantly to this research.
mamíferos y otro tipo de animales grandes
I've also compiled
que
a
usamos
list,
para la investigación, y tenemos una lista de comparación.
though this is by no means a comprehensive list of
Esta es una
all
lista bastante grande
from large vertebrates. What is
con vertebrados grandes
listed here is that they
que
involve
podemos ver acá,
with excellent preservation.
en
So
donde
they
podemos ver una excelente preservación de los restos.
permafrost or cave sediments. The exception to this is
La
the
excepción a esto
Anderson and colleagues 2012 and
es una investigación de Anderson y de Thomas,
2018. Anderson and colleagues 2012 collected soil samples from a
en
number of
donde hemos recolectado información del suelo en áreas ambientales.
while Thomas and colleagues 2018 sampled
Esto
soil.
se realizó en el 2018.
These two studies
Estos dos
examined
estudios
much shorter deposition timeframes.
se hicieron en corto tiempo de descomposición.
All the studies listed here were successful in recovering targeted DNA.
Todos estos estudios se hicieron de forma exitosa al recolectar el ADN
Though it's worth noting that either chloroplast DNA or mitochondrial DNA were the
y
primary.
también se hizo recolección de ADN mitocondrial.
So as I mentioned, I also have experience related to the recovery.
Entonces, como les mencioné, esta es la persistencia del ADN. Lo hemos
particularly
recuperado
I'll use the next few slides to kind of review my research on this topic, which
y las próximas diapositivas va a ser una especie de revisión de la investigación que hemos realizado todos estos años.
this research was in collaboration with another panelist who just spoke,
También hemos colaborado con otros especialistas en el área
and it used willfully donated human decedents at the anthropology research facility, which she also
y
introduced.
hemos hecho la investigación con donantes de cadáveres que lo hemos realizado. Estas investigaciones las hemos realizado en las instalaciones de la universidad.
So the purpose of this
Entonces,
research
el
was
objetivo
to
de esta investigación
examine DNA in soil.
es hacer el examen de la persistencia del ADN humano en el terreno, en cuanto a la descomposición.
This was done over the course of decomposition by evaluating the presence and quantity of two human DNA targets.
Aquí se hizo
A
una evaluación de la presencia y la cantidad del de ADN existente de dos seres humanos en el terreno.
nuclear DNA target that was 67 base pairs and a
Y aquí se
mitochondrial
hizo
DNA
una investigación del ADN nuclear humano de 67 pp.
To track the persistence of these targets, we placed 4 willfully donated human individuals
Para esto se
at
utilizaron 4 cadáveres
or the
en la
body farm
investigación de la descomposición en la granja
on soil that had not previously included
en
cadavers.
terrenos que antiguamente no habían incluido cadáveres.
The experiment began in 2012.
La
And
investigación empezó en el 2012
were collected beneath the torsos of each individual throughout
y
decay.
recolectamos los torsos de cada uno de estos individuos.
And I will kind of be referencing decomposition stages throughout, which I've listed here.
Aquí
And
tenemos las muestras y las etapas de la descomposición de
so
los
fresh
cadáveres.
is going to generally refer to like recently placed or recently decomposing remains in the kind of first few days of decomposition.
De la descomposición de los cuerpos, de investigaciones recientes al respecto.
Bloat is more during that stage of putrefaction.
A través de la
Active
etapa de putrefacción
is where
activa,
you're going to see a large
en donde
mass loss.
podemos ver una gran cantidad de microorganismos
And advanced
activos.
decay is when decomposition begins to slow.
Y aquí es cuando la descomposición empieza a hacerse más lenta.
And then, of course, dry remains is basically when you end up with skeletal remains.
Y obviamente al término de este proceso terminamos con un esqueleto.
I'm also going to reference
También voy a
cumulative
hacer referencia a, perdón,
which
no alcancé a escuchar esa parte.
is basically a way that we keep track of time while also considering temperature, since temperature is
Obviamente, a través del tiempo hacemos rastreo de las diferencias de temperatura a través de las distintas etapas.
So initial samples were taken at placement of the individuals, and then the
Las
last
muestras
sample was
iniciales se hacen al principio cuando se coloca el cadáver en el lugar
after death.
y después hacemos todas las siguientes etapas hasta el proceso final de descomposición hasta llegar al esqueleto.
Preliminary PCRs were used to
Los
monitor the
PCRs
or
iniciales
absence of nuclear and
se
mitochondrial.
monitorean para investigar cuál es el ADN mitocondrial y el ADN nuclear.
We then used a quantitative method of PCR, qPCR, to
Luego
quantify
utilizamos el método cuantificado
the base pair mitochondrial DNA target.
para poder obtener ADN mitocondrial.
We used repeat silica extractions to remove any inhibitors, which can be a problem for PCR.
Utilizamos todos aquellos inhibidores que podrían ser problemáticos para la investigación, para que así la investigación sea limpia.
And we sequenced the hypervariable regions 1 and 2
Y hacemos la secuenciación de las áreas 1 y 2
to authenticate DNA. So we
para
compared
autenticar el ADN. Por lo tanto, hacemos comparaciones
to
de
DNA.
la sangre, del ADN.
So ultimately, the
Y
results of this
finalmente,
experiment showed that we were able to
Le
recover
mostramos
human
aquí los
oils.
resultados de lo que hemos obtenido.
Nuclear DNA
Donde
amplified in
tenemos ADN nuclear de las distintas muestras.
This was unrelated to time since death or the individual assessed.
Esto se refiere al proceso de evaluación de los cadáveres en una etapa inicial.
Mitochondrial DNA was amplified in 35 of 41 samples.
El ADN mitocondrial fue amplificado en las 30 o 40 muestras que hicimos.
And following sequence authentication, we removed 2 of the 4 cadavers from any further
Y luego
assessment.
hicimos investigaciones con los 2 cadáveres que quedaron al final para poder hacer la secuenciación total de principio a fin.
The main results are pretty much
Los
summarized.
resultados finales están resumidos aquí en estas tablas,
Both figures show log gene copy number per gram of dry weight soil on
en
the
donde
y-axis
vemos el tiempo en el proceso, en el terreno,
and time on the x-axis. So in the first plot,
Lo
samples
primero que
are
vemos acá son muestras
into those decomposition stages that I mentioned.
en las etapas distintas de descomposición que podemos ver, etapa inicial, etapa de hinchazón, etapa de descomposición hasta al final.
While in the second plot,
Y en la segunda
continuous
tabla
and negative degree hours.
podemos ver la información en cuanto a horas en las distintas etapas.
Both figures are basically showing that as
Estas cifras nos indican
per gram of dry weight soil increased as
l-
decomposition progressed.
una información respecto a la cantidad de gramos de terreno seco en cuanto a peso, comparado con la descomposición en progreso,
And then decreased throughout the remainder of time.
y luego todo esto, estas mediciones empiezan a decaer a medida que pasa el tiempo.
An important point to also mention about this study
Un
is
punto también importante que se puede mencionar en este estudio es que
that we were unable to authenticate human DNA sequences from the dry remain stage of
no
decomposition.
pudimos hacer autenticación del ADN humano en la etapa cuando ya estaba seco los cadáveres.
And those samples were collected at days 83, 87, 114, and 198.
Y esas muestras se identificaron en distintos periodos de horas en el proceso de descomposición.
Sequences from these samples
La
were
secuencia de estas muestras, perdón, no la escucho muy bien, se corta la transmisión.
at the time I was using primers that targeted very large amplicons or very large fragments,
Al
around
principio empezamos a utilizar fragmentos muy grandes
and 556 base pairs. So
y
at
con
a later
distintos pares.
time, I tried to use overlapping smaller primer sets and the
Y
sequences.
después, a medida que se producía la descomposición, obviamente empezamos a utilizar piezas más pequeñas para poder realizar las muestras.
So from this experiment,
Entonces, de estos experimentos, las conclusiones
we
y
learned
las
that we
limitaciones,
mitochondrial DNA from soils associated with cadavers
podemos
that,
recuperar ADN mitocondrial a través del proceso de descomposición,
but unfortunately we were unable to consistently amplify nuclear DNA, which would be
pero
ideal for
lamentablemente no pudimos hacer amplificación del ADN nuclear.
moreover, cadaver mitochondrial profiles could not be authenticated in the experiment beyond 48 days, which is a
Más allá de
relative.
los 48 días, no se pudieron hacer secuenciaciones del HBI o del HBI2.
The experiment could definitely be improved. First, we could include
El
more,
experimento se puede mejorar, obviamente. Podemos incluir nuevas pruebas
and which would basically allow us to better understand how long DNA persists and the quality
Esto nos
over time.
permitió saber cuánto tiempo permanece el ADN, perdón, de forma estable para poder hacer una identificación correcta.
Second, an improved study design could include more sensitive methods that could be
También
used.
tenemos que tener otros métodos que sean más precisos para poder hacer la identificación correcta.
Next generation sequencing in particular could be used to help us recover nuclear DNA instead of mitochondrial DNA.
Para las próximas secuenciaciones, para la próxima generación, podemos encontrar y utilizar el ADN nuclear en vez del ADN mitocondrial, que es mejor
This
para el
could
proceso
also
de
be
investigación.
combined with something like targeted enrichment,
Eso se puede hacer a través de un método enriquecido de objetivos,
which allows us to basically target regions of interest instead of sequencing everything. And then
de tal manera que podamos dirigirnos solamente a algunas regiones específicas en vez de hacer una secuenciación de todo.
another option is to use something like forensic DNA signature prep, which also takes
También
advantage
podemos utilizar un sistema de forense de secuenciación
and uses these targeted baits. So that something like that.
utilizado según los objetivos que podamos establecer para la investigación.
However, at this time, more testing is needed to
De todas
definitive
maneras, necesitamos realizar más pruebas para poder entender qué tanto
sorry,
tiempo permanece de forma inalterable el ADN humano en
and
el
in what
ambiente.
So beyond our research designed specifically to assess
Aparte
existence
de
of
esto, tenemos algunos otros proyectos de recuperación de ADN
eDNA recovery projects, generally in collaboration with the National Geographic Society or funded by the National Geographic Society.
en
So I'll just-
colaboración con el National Geographic Society.
First, I'm currently working on a
En lo que
project
yo estoy trabajando ahora en este momento es en un proyecto
from cave sediments collected in Portugal.
en recolección de sedimentos en una cueva en Portugal.
This is from a cave. The goal
Esto
is
es de
to
una cueva.
understand more regarding the timing of occupation of Neanderthals and anatomically modern
Esto
humans,
tiene que ver con la ocupación de neandertales en esa época, recuperación de información de mamíferos,
especially in the absence of any other biological material in the cave.
obviamente, si es que no existe otro tipo de material biológico en la cueva que podamos utilizar para la investigación.
So far we've collected sediments from 15 areas around the cave.
Ahora hemos recolectado más o menos unas 15 muestras de distintas áreas en la cueva
Spanning soils dating between 30 and 40,000 years ago.
y estamos hablando de zonas de 30 a 40 mil años de antigüedad.
DNA was extracted using a magnetic bead method, which was optimized
Utilizamos
for
un método magnético para poder recuperar el ADN.
we've basically done 3 extractions per every soil sampling location and
Y
included
hemos hecho 3 extracciones por cada una de las ubicaciones de las muestras.
we used the targeted enrichment that I mentioned on the previous slide
Hemos
to
utilizado el método de enriquecimiento del objetivo que ya les mencioné.
DNA. And in those targeted baits that we use, we also
Y en
include
estos
humans and Neanderthals.
ejemplos hemos incluido como objetivo los humanos del Neandertal.
Preliminary results do point to possible ancient human or Neanderthal DNA in at least a couple of the samples. But
En
of
alguna de
course,
las muestras que tenemos es posible encontrar resultados preliminares que apuntan a humanos antiguos o neandertales. Ese es el ADN que se ha obtenido.
more work is needed to be done to authenticate any recovery sequences.
Pero obviamente se necesita realizar más trabajo para poder autenticar todas estas secuencias recuperadas.
I also want to point out with this kind of work, the
Como ya les he
possibility
señalado, con respecto a este tipo de trabajos existen posibilidades de seguir avanzando,
especially when trying to amplify human DNA, because we're humans
especialmente
are.
cuando se trata de hacer una amplificación del ADN humano.
So you can see when these samples were collected, I was wearing full protective equipment here, so that
Obviamente aquí yo para poder recolectar las muestras tengo que estar protegida para no contaminar el área.
any molecular work then has to be done in clean labs or
Y
labs that
el proceso de recuperación del ADN se ha realizado en laboratorios nucleares, obviamente totalmente asépticos.
the second recovery project that I want to
El segundo
mention
proyecto en el cual estoy trabajando
missing person investigation on a tropical island.
Esta es una investigación asociada al terreno de una persona que desapareció hace 80 años en una isla tropical.
This recovery project was a long shot, not only because of the tropical conditions, which weren't conducive for DNA preservation, but also because
Obviamente, esta investigación es un tanto más compleja por, obviamente, las condiciones ambientales, porque estamos hablando de una zona tropical,
we didn't actually have a known burial site or a known site of decomposition.
y también por el hecho de que no tuvimos una área específica de descomposición del terreno, como para poder investigar en forma más específica.
The sampling was actually based on areas that were associated with cadaver dog alerts. And the dogs,
Las áreas de investigación básicamente fueron áreas que fueron detectadas por perros que buscaban cadáveres.
though we tested multiple extraction methods
Por lo
and also
tanto, utilizamos distintos métodos de extracción para poder
next gen
abarcar una amplia gama de posibilidades,
we were unable to recover mitochondrial DNA.
pero no fuimos capaces de recuperar ADN mitocondrial.
So now I'm going to basically discuss how
Ahora
this research
básicamente vamos a enfocarnos en cómo esta investigación se puede aplicar en Colonia Dignidad.
the ability to recover human DNA of high enough quality is dependent on environmental conditions and
Obviamente
technical.
las condiciones ambientales son aquellas que predominan al momento de tratar de recuperar el ADN ambiental.
The persistence of human DNA in soils will ultimately depend on environmental conditions.
Por lo
So
tanto, las condiciones ambientales son prevalentes al momento de recuperar el ADN.
DNA will begin to degrade immediately following death during
Obviamente,
autolysis.
después de la muerte, de forma inmediata, el ADN empieza a degradarse.
And then it will continue through putrefaction, which is bacterial
Luego
degradation.
continuamos con la degradación bacterial a través de la putrefacción.
The DNA will fragment through
El ADN se fragmenta.
and extracellular nuclease activity, which are enzymes that are going
Distintos
to
tipos
degrade.
de actividades celulares.
And then will also degrade spontaneously
Y
through
también se
hydrolysis.
degrada de forma espontánea a través de la hidrólisis.
High temperatures, increased UV, low soil pH, high moisture, and increased microbial activity are all bad for preservation.
También parte del proceso son la radiación UV, el calor, el tipo de terreno, los substratos, las áreas húmedas, el pH, la humedad y la actividad microbial.
To survive in the soil, DNA basically has to escape any mechanisms of degradation.
Obviamente, para poder preservar el ADN tendría que preservarse de cualquier tipo de degradación ambiental, como las ya mencionadas,
Which can occur by absorbing to the soil.
lo cual puede suceder cuando es absorbido por la tierra.
So different soil types can absorb DNA at different efficiencies. So sand,
Obviamente, los
clay,
distintos tipos de terrenos pueden absorber en distintas medidas el ADN dependiendo de las condiciones del terreno
as well as different organic compounds in the soil, such as humic
y
acid.
también dependiendo del tipo de acidez y del tipo de compuestos orgánicos que existen en el terreno.
But this binding of the DNA can also make DNA harder to recover and analyze due to things like inhibition in PCR.
Obviamente, debido al PCR o debido a la inhibición del ADN, podemos tener menor posibilidad de obtenerlo a través de la detección en terreno.
So not only would the preservation need to be good to recover
Por lo
DNA,
tanto, la preservación del ADN no solamente tiene que ser buena, sino que también el tipo de recuperación, el proceso de recuperación también tiene que ser excelente.
but we would also need to use sensitive enough methods to get the
Y
information.
también necesitamos tener métodos adecuados para obtener la información que necesitamos.
DNA extraction methods would need to be optimized to increase recovery.
Tenemos que optimizar los métodos de extracción para mejorar los resultados.
In the study at the body farm that I mentioned, only 2 to
Y
7,
los estudios de la granja de cadáveres, perdón, esa parte no logré escucharla.
which is incredibly low.
El proceso es bastante lento cuando se trata de hacer investigación en este tipo de granjas.
And if possible, we would also want to be able to extract nuclear DNA, not just
Y también
mitochondria.
deseamos obtener ADN nuclear, no solamente mitocondrial.
So recovering from DNA from graves also has its own
Entonces,
set of
la recuperación del ADN de algunas tumbas, esto también es algo que podemos hablar.
There can be extensive bioturbation in soils or mixing up of the soils due to vertebrate and invertebrate access to the soils.
Debido al acceso de algunos vertebrados e invertebrados se produce una perturbación del terreno y una lixiviación también de los productos en descomposición.
This would cause materials to move around and increase the chances of leaching.
Por lo tanto, esto al moverse el terreno se produce una mayor lixiviación.
There are also sampling challenges. For example, decomposition materials likely will pull to the base of a grave.
También obviamente hay algún tipo de materiales que se descomponen y que se van a la base de la tumba, que son más difíciles de poder recolectar.
Though there has been a study that was able to amplify pig DNA above carcasses in burials, it's
Aunque
unclear
también
whether
hemos realizado algunos estudios con cerdos en donde sí hemos podido detectar el ADN a pesar de que ha pasado bastante tiempo en la tumba,
more work needs to be done
pero se
to
necesita
understand
realizar más investigación.
leaching. In addition, any person present at the site or collecting soils could inadvertently contaminate the soils.
Obviamente también cuando hay presencia de personas que están en el área, también eso significa que hay una contaminación del terreno para poder hacer la investigación posterior.
And for example, in this case, if remains were disinterred, the soils would then be disturbed and mixed, and the chances
Obviamente si se produce el desentierro del cadáver, ahí se produce una perturbación del terreno en donde implica que no podemos obtener los resultados óptimos.
I'm not sure whether we're actually discussing single graves, mass graves, or both in this case.
En este caso podemos hablar de tumbas individuales o colectivas,
As the decomposition materials would mix, which would lead to
porque si es
mixed.
así, cuando son mezcladas, obviamente se mezcla el material de ADN.
These would be difficult to disentangle.
Por lo tanto, es más difícil de poder revisar exactamente cuál es el ADN de un individuo o de otro.
So eDNA as an approach for forensic identification, though a long shot in general, would be more appropriate for single burials.
Por lo tanto, el tipo de identificación de ADN es más apropiado cuando se realiza en tumbas individuales.
And again, more testing needs to be conducted to actually understand human DNA persistence and preservation before the approach is really field ready.
Y obviamente para poder sentirnos realmente confiados en los procesos, tenemos que realizar más investigaciones respecto a la persistencia del ADN y se necesita realizar más preservación de cadáveres también para realizar más investigaciones.
I've listed some other avenues of research here at the
Aquí
bottom.
hay parte de la investigación que hemos realizado que ustedes también pueden revisar.
I think we're probably out of time, so I'm not really going to go
Creo que
into
ya
them.
no tenemos más tiempo, así que bueno, eso es lo que les puedo mostrar hasta el momento.
But again, thank you for having me.
Pero nuevamente, gracias por escucharme.
Muchas gracias, Alex, por la presentación. Creo que fue muy claro, mostrando los desafíos que nos presenta el caso de Colonia Dignidad a nivel científico, donde las cosas, no digo que sean imposibles, pero son bastante complejas, y es por eso que un caso así necesita, como fue el patio 29, una comisión de especialistas de diferentes países del mundo, junto con los científicos chilenos, para abordar el caso como tiene que ser. Nos queda poco tiempo, casi unos 10 minutos. Pido disculpas por los
we have
problemas
little
en el
time. I
mecanismo
apologize for the
de
mechanism
traducción.
used for the translation.
Pero si hay problemas, si hay preguntas, comentarios, podemos avanzar
We can
rápidamente.
continue maybe if we have other questions.
Tenemos una pregunta para Jennifer.
We have a question for Jennifer.
¿Qué se espera encontrar casi 50 años después
What is
para
expected to be
tener
found
evidencias que hubo
to have evidence
cuerpos
that
en el lugar.
there were bodies found in the place. What else do we have?
Could you repeat the question?
¿Puede repetir la pregunta, por favor?
¿Qué se espera
What
encontrar
can we expect to find
casi 50 años después
50 years after?
para tener evidencia que hubo
Have
cuerpos en el lugar?
evidence that there were bodies in that place.
Okay, thank you. Yeah, the length of
Gracias.
time is a big challenge for trying to detect.
Gracias. El tiempo es realmente un desafío para poder hallar evidencias.
I know in other archaeological scenarios, sometimes phosphorus is used as a tracer of the presence of humans because of its concentration in human bone.
Yo sé que en algunos lados en escenarios o lugares donde se hacen búsquedas arqueológicas se usa el fósforo como un índice ya que está bastante presente en cuerpos humanos.
But whether I'm not as familiar with the research on phosphorus and some of that evidence, and so I'm not sure if that would be applicable in this scenario.
Lo que sí es que no estoy tan familiarizada con el uso del fósforo en digamos, y como evidencia, por lo tanto, no sé cómo se podría aplicar en este caso, en este escenario.
Otra pregunta, ¿la humedad, la lluvia y la materia orgánica pueden dañar o favorecer la descomposición? Esto también para Jennifer.
Jennifer, also another question, humidity, rain, organic matter, can that be favorable or unfavorable for the decomposition?
Absolutely. All of those things are very favorable for decomposition. So microbes, like all life, need water. So under more humid, more moisture conditions, you're going to have much faster degradation. Likewise, in scenarios when we have more organic matter, more plant matter, that helps to balance out the nitrogen that's coming in from the bodies, and so that can speed up decomposition as well. The scenario to think of maybe is like composting.
Sí, claro. La humedad y la materia orgánica son todos muy favorables para la producción de microbios, el agua, a su vez la humedad. Entonces, de la misma manera, y esto digamos, aumenta la descomposición, de la misma manera que materia orgánica, plantas, de alguna forma generan un equilibrio y aumentan la presencia de nitrógeno, como lo estaría los restos de un cuerpo. Todas estas son condiciones que también podrían ayudarnos a tener una lectura del tipo de descomposición.
We can think of these scenarios a little bit like how we might think about backyard composting when we compost food scraps. The things that promote decomposition there are temperature and moisture and organic matter. So those things will speed up decomposition in these cases as well.
Como decía anteriormente, que esto podíamos pensarlo que fuera como un compost, incluyendo materia orgánica. Entonces, un escenario, pensemos lo que es nuestro patio, donde hacemos compost y tenemos todo este material, la tierra, la material orgánico, la humedad, genera y nos da una situación específica de descomposición. O sea, miremoslo como de esa forma también.
Ok. Luis Narváez, yo no tengo acceso a las preguntas. ¿Hay alguna pregunta más? ¿Nos queda tiempo para una o dos más?
Sí, Rodolfo Muñoz pregunta si es posible que no existiendo restos de huesos, aún queden evidencias biológicas de un cuerpo que estuvo 5 años enterrado y luego fue sacado, como es el caso de Colonia de Dignidad, y se pueda encontrar ADN.
Mr. Rodolfo Muñoz is asking if it's possible for to find any evidence if there were remains or bones and this biological conditions and they were extracted 5 years afterwards. Is there any case of a way of finding maybe remains of DNA as was the case of Colonia Dignidad?
Are we talking about DNA in bones?
¿Se habla de la ADN en los huesos específicamente?
No. La pregunta dice, ¿es posible que no existiendo restos de huesos?
Sorry. It's possible that if there are no remains of bones. Sorry, that's my apology. There's no remains of bones in that condition. Si puede continuar con la pregunta y aclaraba esa parte.
Okay, just so I'm, just to clarify, we're asking whether we could maybe get DNA in the absence of biological material.
O sea, es encontrar restos de ADN en la ausencia de restos biológicos. Esa es la pregunta.
Esa es la pregunta.
Correct.
It is possible, but like probably not likely.
Es posible, pero probablemente no sea factible. Es posible, pero...
And that all has to do with the conditions in the environment itself, what the soils are like, what the temperature is like, how much water is in the burial, how much microbial activity is there.
Y claro, quizás, pero va a depender de las condiciones y del entorno, o sea, el ambiente, la temperatura, el tipo de suelo, el nivel de humedad, también la actividad microbiana en esa condición.
Tenemos más preguntas, ya son las 4:30. Luis, ¿hay más preguntas?
They're asking if there are any more questions.
Sí, Juvenal Arias hablaba algo similar a lo que acaba de responder Alex, en términos si la humedad, las lluvias y materia orgánica pueden dañar o favorecer la descomposición y que queden, acá dice que queden elementos como para identificar a una persona, a un ser humano.
Ya fue contestada esa sobre el efecto de la lluvia y el material, ya fue respondida.
There was a similar question that was answered on the effects of humidity and decomposition.
Si no hay más preguntas, Paco, ¿querés decir algo?
Un simple apunte. En España hemos abierto unas 700 fosas de hace 80 años y llevamos realizando análisis químicos de la Tierra para intentar comprender por qué en ocasiones el ADN está preservado y en otras ocasiones está degradado. Y todavía no lo entendemos. Pero yo creo que hay que señalar que lo que más degrada el ADN, en general los elementos biológicos, pero particularmente el ADN, son las oscilaciones medioambientales en donde puedan estar esos restos. Es decir, la humedad puede degradar el ADN, pero también lo puede conservar. El problema son los cambios, las oscilaciones. El problema es que ahora haga frío, mañana haga calor. Hoy haya humedad, pasado mañana hay una desecación. Esta es la razón por la cual en cuevas, en Europa, se ha preservado, por ejemplo, el ADN. Porque cuando el ADN está en una envoltura arcillosa y la arcilla actúa como un envolvente que garantiza, digamos, la permanencia idéntica en temperatura y humedad, resulta que el ADN puede durar muchos años, miles de años. Ahora bien, si el envolvente, el estuche en donde está el ADN, la arcilla que envuelve a los restos, experimenta inundaciones, desecaciones y cambios térmicos, entonces hay, en mi experiencia, una mayor degradación. Por lo tanto, se suele decir que el agua degrada, cierto es, pero lo que más degrada es que hoy se seque completamente hasta el extremo y pasado mañana se inunde completamente.
So just to summarize, resumo, just to summarize,
sí, por favor.
In Spain they've had the opportunity of unearth over 700 different remains for different periods of long extended periods of time they've been unearthed. And they've seen that, yes, although there are, and through chemical analysis, different techniques, seen that DNA can degrade, can't really, through biological conditions, they really can't pinpoint out why some DNA remains in some conditions and others are lost, any reading of them. So, but they have detected that the oscillations in the environments where the remains are, in other words, where there's humidity one day and then there's a full total dryness in the next, that is something that degrades or does not enable the permanence of the DNA reading. Now, these oscillations of temperature and the environment, of weather, humidity, and other aspects is something that may lead to understanding better how the DNA can be read. This is one of the reasons why in caves they found that the clay or environment sort of wraps bones around and somehow protects the remains from all these thermal changes. So water can somehow favor or not, but it will depend on these oscillations as discovered. Pues eso.
Muchas gracias, Paco, por el comentario final. Lamentablemente no tenemos más tiempo. Les quiero agradecer mucho a Jennifer, a Alex, a Paco y a Iván por haber dado este tiempo para compartir. Seguramente habrá más reuniones porque este es un problema que hay que resolver porque hay muchos familiares esperando y porque Chile se lo merece. Así que gracias a toda la audiencia y gracias a la Universidad de Chile y a la agrupación de detenidos desaparecidos de Maule. Hasta siempre, adiós.
Okay.
Bye, bye.
Bye.
Diego, ¿terminó la transmisión?